Hi guys, Miss Francis here to talk about another DNA technology called gel electrophoresis, which allows us to do something called RFLIP analysis. RFLIP stands for Restriction Fragment Length Polymorphism. Now, everyone has different sequences of DNA, so if I use a restriction enzyme to cut my DNA, it's going to cut at different restriction sites versus taking someone else's DNA and using that same restriction enzyme to cut it because that other person has different restriction sites. So what you end up with is fragments that end up being different lengths. And when these fragments are used for a technique called gel electrophoresis, the different fragments will migrate in different patterns, forming bands based on their different sizes. So after treating long DNA molecules with a restriction enzyme, the fragments can be separated by size via gel electrophoresis, where it produces a series of bands that are unique to the sample of DNA and unique to the restriction enzyme that's used. So as I said, the separation depends on size based on the length of the fragment, where the fragment is measured in base pairs. The longer fragment will migrate less down through the gel. And let me show you what I mean. This here is the gel, where I've got a power source along with what's called wells located at the top of the gel. It's within these wells that I'm going to put my DNA sample. Then I turn on the power source and my DNA samples migrate down the gel. The reason why they migrate down the gel is because the bottom of the gel is positively charged. Remember, DNA contains a phosphate group, which is negatively charged. So that negative phosphate group wants to move towards that positively charged end of the gel. Where within the gel, the shorter fragments, the shorter molecules are going to travel further down the gel. Now we can use restriction fragment analysis to compare different DNA molecules, for example, representing different alleles, because after all, alleles are different versions of a gene, therefore they're going to differ in their DNA sequence. So they'll probably also differ in their restriction sites. Well, if they differ in their restriction site, well then each allele is going to produce a different sized fragment when digested or cut by the same restriction enzyme. And then it's going to produce a unique or a different banding pattern, which allows us to distinguish the two alleles. So let me show you what I mean. So here we've got, sorry. Here we've got allele 1 and here we've got allele 2. And do you notice that there is a slight difference in their base sequences? However, if we use a restriction enzyme that cuts at CCGG in between the C's, do you see that in allele 1, I have 2 restriction sites, so I'm going to have 2 cuts. But in allele 2, I only have 1 restriction site, so I'm only going to have 1 cut. So when I have 2 cuts, I'm going to make 3 fragments. So here's my first fragment, fragment Y. Here's my second fragment, fragment X. And then here's my third fragment, fragment W. Now, if we look at our second allele also has fragment Y, but our second allele has this larger fragment, fragment Z. So if I have 1 cut, I make 2 fragments. If I have 2 cuts, I get 3 fragments. If I have 3 cuts, I make 4 fragments, and so on and so forth. You guys see the pattern. So if I were to put allele 1 and run it in a gel, this is what the banding pattern would look like, where my small fragment Y is represented by this band. Notice that it has traveled furthest down my gel, closest to the positive end. Then I've got my slightly larger fragment, fragment W, which travels almost as far down, but not quite as far down. And then I've got a band that represents fragment X that's larger than Y and W, so therefore it does not travel as far down the gel as the other two fragments. Now, if I want to look at the banding pattern from allele 2, if that's placed on a gel, do you see that I have band Y at the same location? Because band Y represents the fragment in both allele 1 and allele 2. That's the same size. However, I only have one other band in the gel for allele 2 that represents this fragment Z. So when I compare these two gels, I can see that the banding patterns for allele number 1 and allele number 2 are different. So differences in DNA sequences on homologous chromosomes produce different restriction fragment patterns are found scattered throughout the genome, including the human genome, which means that these RIFLIPs can serve as a genetic marker for a particular location in the genome. So this is in section G of the note packet, but it's applicable right now, where this particular DNA technology offers forensic applications. For example, during a violent crime, blood, semen, or traces of other tissues may be left behind. Well, RIFLIPs and gel electrophoresis can help identify the guilty individual with a much higher degree of certainty than just like a fingerprint, because the DNA sequence of every person is unique, except for identical twins, where even as few as 5 markers from an individual can be used to create that DNA fingerprint, because the probability that 2 people who aren't identical twins having that same fingerprint is extremely small. And then those DNA fingerprints can be used as forensic evidence. So take a look at this evidence below. Here is my defendant's blood. And now here is the victim's blood. Now on the defendant's clothes, do you see how many of the markers found in the DNA fingerprint of the victim are found on the defendant's clothes? The only way that those markers, those bands would be able to show up would be if the victim's blood was on the defendant's clothes. See, the defendant does not have this marker or this marker or this marker, or I shouldn't say marker, really, it's the band. Therefore, it's got to be from the victim's blood. So this technology, DNA fingerprinting, can also extend beyond violent crimes to settle questions of paternity. It can also help identify the remains of individuals killed during natural or man-made disasters. So that's DNA fingerprinting, which involves gel electrophoresis and RIFLIPs.